Hello, good afternoon everyone. So my name's James, I work at eBIC and that's at Diamond Light Source in the UK. To introduce my talk, I'll briefly explain eBIC and what we do and then go through our Cryo-FIB that we offer at Diamond, as well as how to prepare Lamella on grid and CryoET at Diamond, which we offer as a workflow with the FIB. I'll talk briefly about the best practices that I think for Lamella tomography and then go through to subtomogram averaging. So we're funded by the Wellcome Trust, the MRC and the BBSRC, and the aim is to provide expertise for Cryo-EM covering TEM and Cryo-FIB to the user community. We have 5 cryosys, 3 with K3s, 2 with K2s, all with Falcon 3s. We have a Talos and a Glacios. Talos we use for screening and in-house work. The Glacios and one of the cryosys are industry only, and they're offered as essentially as an industry user program. And we have 2 Cryo-FIBs, the SARS and the Aquilos. So this is the SARS and the Aquilos. The SARS has a quorum stage. The Aquilos is Thermo Fisher and has the Thermo Fisher loading system. Oh. So these transfer objects here. Roughly we have about 15 users, of which I would describe 8 as being very active. And we try to offer, generally, we offer about 2 to 3 sessions a week. We have these periods of shutdown where we completely shut down the instruments to do all the maintenance, to make sure everything's running smoothly. And then during the run period, everything works fine. Hopefully everything works fine. I realised that I'm wearing the exact same clothes as in this photo. So for Cryo-FIB-SEM that we offer here, we're doing generally 100% is on-grid Lamella of cells and proteins. And I've offered a typical workflow for the Aquilos. So the Aquilos and the SARS have a different sort of session timetable. Initially, we start with the instrument cooling, the loading of the grid, do the GIST deposition, with the idea that by 3:30, that's when all our fine milling starts, and we start to unload the samples. It's important to note that when you start the fine milling, that's the clock starts then, and you should really get everything done within 1 hour, in my opinion. The idea is that you go home at 5 p.m. Great. I'd briefly like to introduce the commissioning call which we ran. It ended at the beginning of the year. The idea behind this was that we accept proposals, that we then run what we think is a reasonable user program on, to then have an idea of how we offer it to the wider community. We had 3 projects, all of which were looking at cellular material, and these involved 3 8-hour sessions of milling within a 32-hour data collection session on one of the cryosys. I've roughly explained this before, but the main thing for the cryos data collection is before the session starts, you look at all your grids and all your Lamella and you decide which ones are the most important, which ones are the least important, and you start with the most important ones. You also want the one grid which has the most Lamella on, or the largest area to collect on. You want that for your overnight data collection, so you make best use of that microscope. So that was done on the Thursday and Friday, and then again we finish at 5 p.m. on the Friday. Usually there was a session after, in-house session after on the Friday, so the microscopes, the cryosys get used continuously, 24 hours a day. For the commissioning call, generally we prepared 5 rough Lamella, and 3/4 of these were thinned down to fine. So on average, we've got about 4 Lamella per session. My biggest take home was that if the Lamella looks bad at the rough stage, just give up and go on to another position. It's no point making this good. When we transferred it to the microscope, 1 in 10 lamella were lost. Half of them were cracked. They almost all had ice contamination, and this limits the amount of area that you can collect your data from. So in the case of this one here, these chunks of ice, as you tilt over your tomograms, they will come into view and obscure your region. And likewise, you can see a little crack here. So if you have you can't select areas that are close to the crack, there's just too much oscillation and you can't collect data. So something like this would be pretty terrible. There's quite a wide range of contamination levels between lamella. I'm not really sure why that was the case. The things that we learned from the commissioning session: the atlas overview that you get from your screening, which you then bring to me at eBIC. Having this, before the session even starts, you can see where you want to mill. You can see which grid squares were nice, where your cells were. You can even then use this to find where your lamella are in the TEM when it comes to collecting the tomography. Making good notes of what your lamella look like, which sort of areas you have, what your lamella tilt angle is. That's very important. And missing and cracked lamella, in my view, is caused by humans handling the autogrid cartridge. That is, picking up the grid and putting it into the cassette. Keeping that to a minimum, I think, reduces the amount of missing and cracked lamella. So less cracked and less missing lamella, that means more data in the tomography. So after the commissioning, we set up a user program. We offer this as two access methods. The rapid access, which is a single shift or single shifts offered to the proposal. The main point is that you collect the tomography at a different institution. All the proposals are assessed in-house. I technically assess them, and usually the PBSs at eBIC do the biological assessment. But that's all assessed by eBIC. The standard access is a bit more like the commissioning call, where we have 3 days Cryo-FIB, 2 days electron tomography. The proposals are peer-reviewed, so eBIC doesn't really have a hand in accepting those proposals. The main point about accessing the user program is that the grid quality needs to be demonstrated, and this is ice thickness and sample density. It's a single bullet point on one slide in bold. You want your grids to look like this, okay? Cells in the grid squares, lots of cells, the ice looks nice and thin all around the grid. I made this grid. What you don't really want is your grid to look like this. You know, there's maybe just one cell here, there's thick ice all over the central part of the grid. If you put this into the SEM, you might well see nice cells on the surface, but actually below the grid, that's where the ice is, and you can't mill through that thick ice.
Is that your grid also?
That's my grid also, but I wasn't going to say that. So yeah, just to reiterate that ice thickness and sample density needs to be demonstrated. I think the best way to do that is with those TM atlases. You can demonstrate it another way, I just need to be convinced. If you want any information, just email me, I'm happy to answer any questions. The process of on-grid lamella thinning, it was covered a bit by Philip. The idea is that you have these two views of the same region, so this is the same cell here and here. And you can put milling boxes above and below the cell, and these areas, or really these volumes, will be removed in that direction, in the direction of the FIB beam. I've made some animations just to show briefly what the process involves. There's the SEM view and the FIB view, and these are the boxes that hopefully will be removed. I have had problems playing this, so yeah, so the idea is that you remove areas, you expose parts of the cell, you go through different processes, and these boxes move closer and closer together. eventually having a thin lamella going through your cell. The final product in the SEM view looks something like that, and in the FIB view it looks something like that. Just a closer look. You start to be able to see some biological material in the lamella itself, and this part here, this image actually was the previous step to the final milling. I tend to not to image with the ion beam after the final milling is done, but it's just the final thin strip that you see in there. Our standard procedure is that we have an optional sputter coat to coat the grid. We then do the GIS coat. For the SARS, we have about 3 seconds exposure, and for the Aquilos, it tends to be between 4 and 10. The Aquilos hasn't really been optimized fully, and we still get quite a bit of variability in the thickness of that coating. We then sometimes do a sputter coat afterwards. The coarse milling on the FIB, always done at 30 kilovolts. And the lamella tilt angles are generally between 8 and 15. The first step is sometimes omitted if the ice is thin enough. Otherwise, we just step down the current as we move the boxes closer and closer together. The final step of milling, perform at 30 picoamps with the box spacing somewhere between 10 and 300 nanometers. What I do is I start with 300 nanometers and gradually move that box closer and closer together until I can't bear it anymore, and I think I'm going to destroy the lamella. For the SARS, I try to get everything completed, all the fine milling completed within 1.5 hours. For the Aquilos, I really try to keep that to an hour. Occasionally, some of our users do this post-mill sputtering, where you sputter directly onto the lamella, with the coating generally being about 1/8, maybe 1/6 of the initial coating. The CryoET at Diamond. We have 4 microscopes which we run that on. They all have this dual-axis tilt holder, sample stage. The general data collection parameters. When I say general, they're all the ones that I've used have been this. They're either the K2 or the K3. They're all dosimetric grouped tomography with a group of 3 to 10, and they generally go between 40 and 62 degrees tilt. So for my best practices of lamella tomography, you want to minimize the ice contamination from your liquid nitrogen. That means filtering it, that means wearing a mask, trying to avoid any breathing on your sample. If you can, you need to optimize your GIS coat, so that coating is between 1 and 3 microns thick. If you're milling and you see the uniformity of that coating is poor, it just means you need to optimize the process. It's easy for me to say that. I find it very difficult to optimize the GIS. If your lamella looks bad when you start milling, just give up, move on to a different region. You'll save time. Hopefully you can finish all your fine milling within 1 hour. This reduces the ice film contamination that builds up on all surfaces in the FIB. The best way to work is that you prepare lots of lamella on lots of different grids, and then you take them all in one batch to your microscope, and then you do all the tomography. That way you get the best results coming through. It might mean that you spend more time on the FIB, and you possibly might not collect on some of your lamella, but the idea is to make best use of that cryos time. For the tomography, again, just want to reiterate that you need to prioritize which are the best lamella and best grids. The grid with the most lamella you should save for your overnight data collection, so no one has to come in and change grids. And always remember to set your first tilt of the tilt series to your lamella tilt angle. So this means that your 0 degrees tilt is actually perpendicular to your lamella. Just to talk a bit about subtomogram averaging. I'm very new to the field, and I haven't really done any subtomogram averaging. I've tried, but I haven't succeeded. There's a workflow here and a description of the alignment and averaging process from One and Briggs. This review was the best one that I read, and it helped me a lot in understanding the process going on. The point being is, with newer methodology, we're starting to get higher and higher resolution volumes in our tomography. There's this clear trend towards in situ structure determination in cells. We're routinely getting resolutions of less than 8 angstroms, and we're approaching 3 angstroms at the moment. From lamella, you need them to be thin. So Philip was saying less than 140 nanometers. If you can get them thinner than that, maybe that's better. Alignment is key for high resolution. Because you have lamella, you don't have these externally applied fiducial markers. I've had reasonable success with patch tracking, but I still find it's not great. EM clarity, again, I haven't used it directly, but I've been told that it uses the subtomograms as a sort of pseudo fiducial marker to do alignments. So for EM clarity, which you can find more information in Pagin's paper, it uses subtomograms as fiducial markers, 3D sampling for missing wedge compensation, and a principal component analysis classification. And the idea is that you can get slightly higher resolution maps from your tomograms. Another example is from the group of Baumeister. Again, this is prepared from a lamella, so that you can see in the SEM view, in the FIB view, and in the final lamella prepared, you have the you retain the fluorescent signal in that region. And in the segmented volume, you can see the aggregate poly-GA as well as the proteasomes in green here. And what they've done is they can use subtomogram averaging to see the difference in structure between the ground states and the substrate bound states. I also want to introduce a project that is in collaboration with Dave Stewart that we offer at Diamond. And here we have a cell infected with a reovirus, and this is where we've taken lamella from. And the aim of the project was to see the different states of the viral replication within the cell. Again, yeah, great. Okay, so you can see the different the viruses within the cell. This is a relatively low mag, and it shows some biological context. And what the group were trying to do is to pick out the different stages of the virus as it grows. So this is one stage, and this is the early stage. Is these ones that we were interested in. So you can see they have a distinct shape. And the class averages indicate that you have this stage here, and then one of these two. So I think this is just the packed and unpacked virus. Using subtomogram averaging, EM clarity, they were able to get a 7.7 angstrom structure of this intermediate stage. They couldn't prepare purified versions of that stage in particular. And so subtomogram averaging of what was found in the cell was the only way to do it. The lamella were milled at eBIC by Corey. So I wasn't involved in the project as I've shown it. I've just sort of come in after Corey has left to take on later stages of the project. Corey's moved to Stanford. Mark Boyce and Jeff Sutton were involved in the subtomogram averaging, as well as Pagin's group, particularly Ben Himes, with that. So the conclusions are that lamella preparation is starting to be widely adopted, and it's running as a user program at Diamond. the workflows for lamella preparation are well established, and subtomogram averaging can now be used to pick out those in situ structures at particularly high resolution. I just included this again because I really love it. I just like, although it's a low magnification, I like to see all the virus particles as well as the cellular context. I really just like seeing mitochondria. And I just want to thank everyone at eBIC. It makes it a really nice place to work, particularly Corey, who taught me almost everything. So thank you all for listening.
Thank you very much, James. We start discussion. Maybe I should start. Can I ask you this? Very short time you mentioned for fine milling. What is the main reason to be so fast?
So all the surfaces in the FIB, all the cold surfaces in the FIB will grow a film vise over time. It's a fairly constant rate. In our microscopes, we've measured them to be about 30 nanometers per hour. And so if you work for longer than an hour, you're just going to get a thicker film growth. So you get thicker lamella, which obscures regions.
And do you insert 2 grids or 1?
If I have 2, I would work on 2. Yeah.
Okay, thank you. Valentin.
Can you tell us a bit more about the GIS coding optimization? So which steps did you went through?
Okay. So this is generally how the GIS works, is that you sort of spray onto your grid. The green things are cells. The side closest to the nozzle tends to get more of a thicker coat, and the side furthest away. So you can imagine you can change the tilt angle and get a more even coat. You can also get shadowing on cells. For the optimization of the SIOS, so these are 2 different systems, and the optimization of the SIOS was done well before I joined eBIC. It works really well, but I'm not really sure who did it and how they performed that experiment. So I'm trying to optimize the Aquilos at the moment. This is mainly done by changing the distance between the grid and the nozzle. So the further away you move the grid, a more diffuse coating you get put on the surface, but you have to spray for longer. You also start to coat the other grid as well, so it's not great moving away, but if it's the only way to optimize the process. If you mill into the sample and you see it looks like just bubbles in your GIS coating, that suggests there's some kind of gas incorporated into that coating, and you form these bubbles. I've been told it might be water contamination of the GIS, in which case you need to heat up your GIS and vent it into your chamber over about 40 minutes or so. That's what I've done. That's the thing I found best to get a reproducible GIS coating. But yeah, you've got different things to play with, and there's a lot to do.
So you said you also mill crystals, which are relatively high 3D structures on the grid sometimes. How do you get around the problem that actually GIS coating comes from the wrong side for these very large 3D crystals?
Yeah.
Because on Aquilos you basically GIS coat from the back side and you mill from the other side. So actually the front side of your lamella has much less GIS as a protective layer, which is a bit stupid in design, I think. But do you maybe found a good way to get more behind your 3D structure on the grid?
Yes, so I changed the position of the grid. I've been told what you can do is do sort of 3 or 4 separate depositions around your grid, so you get a more even coating. I haven't tried that yet. With the crystals, there tend to be so many good positions to mill that you can actually just get away with a bad GIS coat, and you're going to find some regions which are better to mill. With the crystals, because you have faces, you tend to get more GIS on one face than the other. So if you find a crystal where you're at the edge, you don't want to mill about that edge because one side's going to mill way faster than the other. That's what I find with crystals.
I was wondering about the contamination rates.
Yes.
You said it's 30 nanometers per hour. I mean, it's really significant. How does it compare, or maybe to everyone? It's a question in the fields, and
Yeah.
it's really limited.
Yes, yes it is.
So, what are the general values there?
It's very low compared to what I've seen.
Yeah.
Very low.
But compared to the sequence, it's significant. No?
Yeah.
I mean, 1 hour is nothing.
No, we see 150.
Yeah.
150.
Yeah, 150.
Per hour.
150, that's all.
So just to say that the 30 nanometers per hour, that's an average taken over a test day. And some hours were, maybe 20 nanometers per hour, some were 150 nanometers per hour. So it varies. I think there was a bit of error in our test, and so that might account for a bit of the change in the growth rate. But
Yeah, but it's close to the Lamella stiffness.
Yeah.
No?
yeah,
And altogether, within a factor of whatever.
I think it's a real limiting step for automation processes, and it's something that we need to solve.
But you have a really good one.
A good FIB?
Yeah.
Yeah.
So like saying, we see 150 an hour.
Yeah.
Is there no more questions? We have to move to the next speaker. Thank you very much.
Thank you.